Okay, let's dive into this. To really grasp the difference between forward and reverse mutations, I want you to picture your DNA as this massive, incredibly complex manuscript. Now, a forward mutation, it's basically like a random typo. It takes a perfectly good, active, what we call wild-type word and accidentally turns it into something defective, a mutant. But reverse mutations, which we also call reversions, well, they do the exact opposite. They take that defective mutant sequence and actually revert it right back to the active, wild-type state. It's quite literally nature's backspace key.
And this brilliantly illustrates that a reverse mutation isn't always a perfect, clean backspace, though. See, a true reversion restores the exact original sequence. It perfectly fixes the typo so the protein codes for the exact same wild-type amino acid. But we also have what are called partial reversions. This is where the sequence is changed right at the site of the original mutation, but to a completely different amino acid. The catch is, this new amino acid still fully or partially restores the protein's function. It's kind of like using a synonym that still makes the whole sentence make sense. And then, oh man, then there are suppressor mutations. Instead of even trying to fix the original typo, nature just goes ahead and adds a clarifying sentence somewhere else entirely in the genome just to compensate for the mistake. How cool is that?
Section 1. Spreading the genetic code. So, how does a single fixed typo, or even a brand new forward mutation, actually survive and spread out in the real world? Let's bridge this over to the macro level.
Well, let's look at bacteria, for example. In bacteria, genetic recombination absolutely does not happen through traditional reproduction. Instead, entirely new bacterial strains are produced through three distinct processes of gene transfer. Those are transformation, transduction, and conjugation. Basically, these are the three primary tools bacteria use to swap and spread their genetic code around.
Now, what's really interesting about this is that first process, transformation. Picture a bacterial cell just hanging out in its environment, and it's surrounded by these fragments of free-floating, naked DNA, maybe stuff left over from another cell that died. Through transformation, this living bacterial cell can literally absorb that loose genetic material right out of its surroundings. It just takes it in and seamlessly integrates it right into its own chromosome ring. It's essentially scavenging discarded genetic code and just making it its own.
But here's the thing. Whether a bacterium is absorbing naked DNA from its environment, or maybe getting genes injected by a virus during transduction, or even when human chromosomes cross over during meiosis, the ultimate mechanism at play in all of these is recombination. Recombination is simply the combining of DNA from two individuals into a single genome. And let me tell you, it is the absolute engine for generating new combinations of alleles.
Section 2. Mutations in populations. Because we have to ask, what actually happens when we zoom way out from just a single bacterium or a single recombination event? Let's look at those ripples across scales.
So there's actually a pretty stark contrast between spontaneous mutation rates, and by that I mean mutations that just happen naturally without any outside exposure. If we look at eukaryotes, complex organisms like us humans, we see a germline nucleotide substitution rate that results in roughly 30 brand new inherited mutations per parent. But then compare that to prokaryotes, like bacteria. Their rate is roughly just one mutation in a million cell divisions for a typical gene. These spontaneous, totally random rates, they are the steady drumbeat of evolutionary change across all populations.
However, sometimes a mutation doesn't just sit around waiting to be passed on by statistical chance. And this brings us to a wildly fascinating concept, the selfish mutation. Take achondroplasia, for instance, which is a common single-gene disorder. It's caused almost exclusively by a highly specific, totally targeted nucleotide change at just one single spot in a specific protein. What's absolutely mind-blowing here is that this mutation actually promotes its own transmission. The altered male germline cells gain a proliferative advantage, which basically means they make a disproportionate contribution to sperm. So even with a normal baseline mutation rate, this selfish little mutation practically guarantees its own spread. It's wild. On the flip side of that, some forward mutations get trapped in what we call runs of homozygosity, or ROH. In populations that have high rates of inbreeding over long periods of time, individuals can actually inherit two completely identical genomic segments from a common ancestor. Because the normal genetic shuffling of recombination is severely reduced between highly related parents, recessive disorders can surface way more easily. It effectively traps forward mutations within a population, and as you can guess, that can lead to some pretty significant biomedical consequences. Section 3. Evolution at molecular levels. Let's move to this and see how this builds into genomes, acting as historical records of evolutionary time. We can really contrast historical evolutionary theories by looking at something like bacterial resistance to a virus. In the past, there was this older, adaptive, or Lamarckian idea. This theory suggested that organisms somehow purposefully directed their own mutations specifically to adapt to an environment. But today, we know the accepted reality is random mutation. Genetic changes happen purely by chance, just like those spontaneous forward and reverse typos we were talking about earlier. And occasionally, just occasionally, those totally random changes just happen to be adaptive and give an organism a survival edge.
And we know for a fact it's a random process because our genomes are literally littered with history and mistakes. Scientists used to call vast stretches of our DNA junk DNA, but it turns out a lot of that so-called junk actually produces functional regulatory RNA. On the other hand, though, we do have what are known as pseudogenes. These are basically broken, non-functional copies of once useful genes. You can think of them as the evolutionary rough drafts and abandoned sticky notes left behind in our genetic manuscript.
But duplicating genes isn't always just a dead end. In fact, it creates multi-gene families. A really classic example here would be the globin gene clusters, which make up the oxygen-carrying hemoglobin in our blood. Through gene duplication, an ancestral gene basically just makes copies of itself. And this is a brilliantly clever evolutionary mechanism because it allows nature to safely experiment. One copy can keep doing its vital necessary job, while the extra duplicated copies are free to undergo random forward and reverse mutations. Over long stretches of time, they can potentially evolve entirely new biological functions.
Section 4. Modern genetic applications. Because the real question we have to ask ourselves now is, how does our understanding of these deep genetic mechanisms actually impact our survival right now in the present day?
The source material for this explainer presents some very stark, pressing realities regarding the current food security crisis in Africa. According to UN estimates, 20% of Africa's booming population currently faces severe undernourishment, and this crisis is incredibly complex. It's heavily exacerbated by a web of compounding, real-world factors, including climate change, widespread poverty, conflict, and of course, the massive global disruptions that were caused by the COVID-19 pandemic.
So what tools do we actually have to address a crisis of this scale? Well, the current landscape of genetically modified organisms, or GMOs, offers some targeted opportunities. Things like increased crop yields, engineered pest resistance, and enhanced nutritional profiles. So the crucial point is that this scientific potential is contrasted directly with the reality on the ground, which is a remarkably slow rate of public acceptance and adoption. There's a profound ongoing debate here, constantly balancing the scientific opportunities of biotechnology to address hunger against real public safety concerns and broader societal hesitation.
Which brings us to our final provocative thought for today. We started this explainer by looking at exactly how nature edits itself, from a completely random forward mutation in a single bacterial cell all the way to the complex reverse mutations that literally hit undo on genetic errors. But now we're facing the deliberate genetic modifications required to potentially feed an entire continent. So how will we guide our own genetic future? Our understanding of genetics has finally given us access to the source code of life itself. But ultimately, it's up to us to figure out how and maybe even if we should write the next chapter. Thank you so much for joining me today. Keep learning, stay curious, and I'll catch you in the next explainer.